So in this video, we're going to talk about the spread of malaria between humans. Now, malaria is a life-threatening disease, and it's typically transmitted through the bite of an infected adult female Anopheles mosquito. So an infected adult female mosquito carries the Plasmodium parasite, and when the female mosquito takes a bite of a human, the parasite is then injected or released into the bloodstream of the human. Now, the actual pathogen itself is called Plasmodium. So this is the unicellular organism that causes malaria in a human, and it's, like we just said, transmitted through the bite of an infected adult female mosquito. So we can see here, this is a zoomed in picture of Plasmodium, and it is a unicellular organism. Now, if you actually look inside the Plasmodium, you can see membrane-bound organelles. So we got here a nucleus, and inside that nucleus would be linear DNA. You can also see mitochondria and other structures such as microtubules. There's also ATS ribosomes, where we know protein synthesis occurs, translation. So we know this cell is eukaryotic because of the membrane-bound organelles. So that would make this Plasmodium parasite a protoctistin. So it belongs to the kingdom Protoctista. Now, a unicellular Protoctista is called a protozoan. So Plasmodium is actually a unicellular protozoan pathogenic organism. And this is the one that causes malaria in humans. Now, there's 5 main species of Plasmodium that causes malaria in humans. And the most common one that causes about 50% of malaria cases is called Plasmodium falciparum. Now, the vector shown on the right hand side here is the adult female mosquito called Anopheles. Now, when she lands on the skin of a human, she's got a structure called a proboscis, and that injects into a capillary in the skin of the human. And she takes a meal of blood, so she kind of sucks up the blood, and she'll use that. It goes into the stomach of the mosquito, and she absorbs some of the amino acids and the sugars, etc. But the Plasmodium parasite is located in the salivary glands of the adult female. So while she's taking a meal, these actually also get injected into the blood of the human. And one of the first cells that Plasmodium infects within the human are liver cells. Now, from the liver emerges a slightly different version of Plasmodium that can then infect red blood cells. So one of the symptoms of having malaria is that you get anemia because of the infection of your red blood cells. Now, it's the adult, so the adult female mosquito that is the vector. Now, mosquitoes do not show any ill health by carrying the Plasmodium unicellular pathogen. And the reason that the adult females are the vectors for the malarial parasite and not the male mosquitoes is quite simply that the male mosquitoes do not feed off human blood. Male mosquitoes actually feed off liquid sugar water or nectar that you find in certain species of flower. Now, the adult female mosquito does feed off human blood because there's a higher concentration of protein within that human blood compared to nectar. Now, the actual vector, the female mosquito, she makes eggs. So the life cycle of the mosquito, so we're not talking about the Plasmodium here, but the actual vector is typical insect life cycle. So there is internal fertilization of the female's eggs with the sperm from the male. The female mosquito will then lay her fertilized eggs on stagnant water, so ponds or lakes. And then what happens is they develop into larvae, and the larvae then develop further into new mosquitoes, and they'll fly off, and the adult females will take a meal off humans, and that can increase transmission of malaria in this population, in this area. Now we can talk about the life cycle of the Plasmodium in a bit more detail. So we know it's the adult female who transmits the Plasmodium pathogen into the blood of a human. Now, once these cells get into the bloodstream of the human, they're known as sporozoites. Now, these are haploid cells, Plasmodium cells. Now, Plasmodium has 14 chromosomes inside its nucleus. So haploid simply means one copy of each chromosome. So these haploid sporozoites have glycoproteins on their surface, and when they pass through the human circulation, they can recognize or bind to hepatocytes or liver cells. Now, when they bind to the surface of a liver cell, they infect the hepatocyte. So the sporozoites end up inside the hepatocyte cell or the liver cell. We put liver here, liver cell. Now, inside the liver cell, these sporozoites divide by mitosis. So genetically identical cells are being made, all haploid, so all with 14 chromosomes inside their nucleus. And this structure is called a hepatic schizont. Now, you don't need to know these names necessarily for the exam, but you do need to know that haploid sporozoites will first infect liver cells, and then they divide by mitosis inside them. So we get many, many copies of genetically identical sporozoites. Now, this structure will eventually rupture. So we can see here the schizont has ruptured open, and that releases these haploid Plasmodium cells back into the blood circulation. But this time they've got a slightly different name. It's called a myrizoite. So it's a slightly different version of the Plasmodium parasite. Now, these myrizoites can actually now bind to the cell membrane of erythrocytes or red blood cells, and they infect the red blood cell. So once these myrizoites infect a red blood cell, they can start to divide again by mitosis. We can see here different stages of development, and we end up with what's called a blood stage schizont. So this is a red blood cell that's been infected with Plasmodium. It's replicated by mitosis, and these will then rupture like we saw before with the liver cells, and more myrizoites get released, so more and more red blood cells get infected. So we can see the numbers of Plasmodium cells are going to increase rapidly in the blood of a human. The number of red blood cells is going to decrease at pretty much the same rate, because they're the ones being infected and being burst open. So all these myrizoites are, as we've just mentioned, all haploid. Now, some of these myrizoites undergo what we call differentiation or specialization into what are known as gametocytes. So not all of the myrizoites will do this, just some of them. Now we have a male and female gametocytes, and they are both haploid. So again, they've both got a nucleus with 14 chromosomes inside. Now, a second adult female mosquito will come along, and she will take a bite of the human blood, the same obviously infected human, and infected red blood cells plus myrizoites, Plasmodium cells, plus male and female gametocytes get taken up into the second female mosquito. Now, inside the stomach of the second female mosquito, the adult male and female gametocytes develop into male and female gametes, and they fuse together at fertilization. So fertilization occurs in the stomach of this mosquito, and we get a diploid. We say diploid zygote. Now, this diploid zygote will undergo, so it's got 28 chromosomes now inside its nucleus, several different stages, but then it undergoes meiosis, and this will form new haploid cells that migrate to the salivary gland of the second female mosquito, and they are what we started with earlier, called sporozoites. So the second female mosquito will take up myrizoites plus infected red blood cells plus gametocytes. The gametocytes will develop into gametes. The The gametes fuse in the stomach of the second female mosquito. So that's a diploid cell. They undergo meiosis to produce many haploid cells. These haploid sporozoites migrate to the salivary gland of the second adult female. And that female will then fly off. She'll bite another possibly non-infected human, and she will transmit the sporozoites or plasmodium cells into the second human. So that's the life cycle of Plasmodium. So we've just talked about the life cycle of Plasmodium. Now, we don't necessarily need to know all the specific names of the different stages of the life cycle of Plasmodium, but we do need to know a little bit about the ploidy of these cells. Now, that simply means, are the cells haploid or are they diploid? So we know haploid means one copy of each chromosome, whereas diploid means two copies of each chromosome inside the nucleus of that cell. So if you want to stop the video, you can have a quick go at this activity. All we want in each box next to the yellow number is haploid or diploid. Okay, so number one is the sporozoites, top right hand side here. Now we know the sporozoites are the type of Plasmodium cell in the salivary glands of the female adult mosquito. They get injected into the blood of a human. Now these cells are haploid, and that means inside each sporozoite, in the nucleus of each cell, there are 14 chromosomes, and they are all different. So there's one copy of each of the different types of chromosomes. Now we know these sporozoites, they infect liver cells or hepatocyte cells in the human, and they undergo cell division, and that forms a schizont, and that ruptures open, and it releases a slightly different version of the Plasmodium called merozoites. Now we know that if these sporozoites have divided by mitosis, they're going to make genetically identical cells. So the ploidy remains the same after mitosis. So in number two, we also know the ploidy of these merozoites must be haploid. So inside the nucleus of each merozoite, there are again 14 chromosomes, and there's only one copy of each chromosome. Now again, merozoites infect red blood cells, and inside the red blood cell, they divide by mitosis. So we get genetically identical cells being produced again. If the red blood cell ruptures open, called a red blood cell schizont, if that ruptures open, it releases the merozoites into the bloodstream, and more red blood cells get infected. So these merozoites are also haploid. So again, we know there's 14 different chromosomes inside the nucleus, and there's one copy of each, so they must be haploid. Now, merozoites continue to divide by mitosis. Some of them will differentiate into what we call, we can put differentiate, gametocytes. And we know the gametocytes, because they've been formed through mitosis, they again must be haploid. So we can put 14 chromosomes inside the nucleus there again. We know gametocytes are taken up into the stomach of a second female mosquito. Now inside the stomach, they differentiate somewhat further into actual gametes. So some will be male gametes and some will be female. So the male gametocyte develops into a male gamete, and the female gametocyte develops into a female gamete, and they are both haploid. So once again, 14 chromosomes inside each nucleus. Now inside the stomach of the adult female mosquito, we get fertilization. The male gamete will fertilize a female gamete. The two cells fuse together. It's called an ookinet cell. So that zygote we know is diploid this time. So its nucleus will have 28 chromosomes. So this time we've got 14 different chromosomes, but we've got two copies of each, which makes 28. That further develops into an oocyst cell. You don't need to know the names of these cells, so don't worry about that too much. So again, that will be a diploid cell with 28 chromosomes in its nucleus. Now this diploid cell will undergo meiosis. Now we know meiosis takes a diploid cell and it actually produces 4 genetically different haploid cells. So these sporozoites are actually formed through meiosis, and we know they have 14 chromosomes inside them. So Plasmodium has a sexual life cycle whereby the male gamete and the female gamete in the second female mosquito will fuse together. It forms a diploid zygote that undergoes a bit of differentiation. These cells then undergo meiosis and we get genetically different sporozoites being formed. These sporozoites then migrate to the salivary gland of the second mosquito, and these will then get injected into another human, and the life cycle will continue. So we know a little bit about the life cycle of Plasmodium, but we also need to know some of the preventative measures of the Plasmodium being transmitted from human to human and therefore causing malaria. So there's 2 main types of preventative measure. One is by responding to the behavior of the adult female mosquito and looking at ways to limit the life cycle of the mosquito. And a second method is called biological control. Now the first method we'll look at in a bit of detail. So why would a preventative measure be to sleep under nets? Well, we know the adult female mosquitoes feed at night time. So if you sleep at night time, obviously being under nets prevents the mosquitoes getting onto the skin. Quite often we treat the nets with an insecticide. Now here the name gives us a clue as to why we do that. So pyrethroid is an insecticide, and the adult female mosquito is also obviously an insect. So this insecticide kills the adult female mosquito. Now we know mosquitoes are insects. They actually belong to the arthropod phylum of animal. So this pyrethroid will actually kill the female mosquito, and if she's a carrier of the Plasmodium, then also obviously the Plasmodium is not going to be transmitted to another human. Now you might want to spray indoor walls with the insecticide as well, because obviously after a feed the female mosquitoes tend to rest on the walls, so here they'll be subjected to the presence of insecticide as well. We could drain or cover stagnant water. Now we know the adult female, when she's got her fertilized eggs, she will lay them on the surface of the stagnant water, and the fertilized eggs then develop into larvae that obtain oxygen from the water. So if we drain these ponds or water tanks, etc., we actually remove the egg-laying sites of the adult female mosquito. Likewise, we could actually put a film of oil on the surface of the water, because even if the female mosquito did lay her fertilized eggs, any larvae that started to develop could not pierce the surface to obtain the oxygen they need for further development. So these larvae would actually die off, and that prevents more mosquitoes emerging and being formed. Now, biological control then. Why would fish be introduced into stagnant water like lakes or ponds? Well, these fish would actually eat the larvae, because we know the larvae of the adult female are aquatic. They obtain the oxygen from the water, and the fish would actually use them as a food source. So if there's less larvae, we know there's less mosquitoes going to be formed. Now we could actually infect the adult female mosquito with a particular type of bacterium that is pathogenic to the development of the Plasmodium inside the female mosquito. So these bacteria actually block the Plasmodium from developing inside the female mosquito. So it is technically a pathogen for the Plasmodium organism itself. Now what we could also do is to sterilize the male mosquitoes with X-rays, and this means the males don't make any fertile sperm cells. You can also now genetically modify male mosquitoes to make them infertile. So we then release these into the wild, and they will mate with adult females. However, the adult females will not have their eggs fertilized because the males are sterile, and so when she lays the eggs, they will not develop into larvae, and so no new mosquitoes will be produced. If there's no new mosquitoes being produced, or less of them, then the transmission of the Plasmodium parasite is also going to be less. Now, one of the benefits of doing this last point here, is that if you sterilize male mosquitoes, they will fly off in the wild and they will actually find the females to try to mate with. So it prevents the scientists from having to go out and actually locate and find all of the adult female mosquitoes themselves. They kind of let the male mosquitoes do the job for them. So, drug treatment then of malaria. Well, vaccines do exist for malarial parasites, but they're not necessarily that effective. Now, one of the reasons is that the parasite, the Plasmodium parasite, actually has quite a high mutation rate. We know it reproduces sexually as part of its life cycle, but also it has quite a high rate of mutations occurring. So quite often we get different antigenic types of Plasmodium, to which a vaccine might not be that effective. So if you wish to have a quick go at this quiz, you can maybe pause the video, jot down some answers, and then I'll put the actual answers on the next slide. Okay, so if you look at the first question then, what is the infectious pathogen that causes malaria? Well, we know it's Plasmodium. Now, Plasmodium falciparum is the actual main species of Plasmodium that causes malaria, about 50% of cases in humans. We know Plasmodium belongs to the Protoctista kingdom. It's a protozoan. It's a unicellular eukaryotic cell. Now, three characteristics of this type of organism then. Well, unicellular, eukaryotic. We know if it's eukaryotic, the nucleus must have linear DNA. So it's very different to bacteria that have a circular DNA inside the cytoplasm. And we also know they're heterotrophic in this particular case. Now, what is the vector and the primary host that transmits the malarial pathogen? It's the adult female mosquito. So the adult female mosquito is the vector, not the male. Now, the secondary host is obviously the human. Inside the secondary host, what two cell types does the pathogen replicate inside? Well, we know initially it's liver cells, so these release merozoites, and the merozoites then go off and infect red blood cells, otherwise known as erythrocytes. Now, in the blood plasma, the pathogen can differentiate into either male or female gametocytes that further develop into the male or female gametes. These get transferred to a new primary host when it feeds on human blood. Where does fertilization occur in the primary host, and where does the zygote then migrate to? Well, the zygote forms inside the stomach of the second female mosquito. It further develops and moves or migrates towards the salivary gland of the mosquito. So in that salivary gland, you end up with more sporozoites, formed by meiosis of the cell that developed from the diploid zygote. So sporozoites, we know, are haploid. Now, what would be three preventative methods for reducing the spread of malaria? So you can either target the Plasmodium itself, or you can target the adult female mosquito, which is the main method. So we could sleep under mosquito nets, we could cover the nets in insecticide, we could actually cover the walls of the room that you're staying in with insecticide too. The male mosquitoes could be sterilized with x-rays, or they could be genetically modified to be sterile and then released back into the wild. So we could say here, released back into the wild. Because we know obviously the female's eggs need to be fertilized in order for those eggs to develop into larvae. We could drain stagnant water such as ponds, streams, lakes, puddles, or we could cover the stagnant water so the female has no access to egg laying sites, or we could even place a film of water over the a film of oil over the water, because we know larvae pierce the surface and that's where they obtain their oxygen from. So if you prevent this occurring, you prevent development of the larvae into new mosquitoes.